Well, good afternoon, everyone, and welcome to the June e-learning series webinar presented by Temple Health and Bioscience District. Before we get into today's program, I'd like to thank you all for tuning in and tell you a little bit about THBD. Big things are happening in the Texas life science industry, and Temple is one of the fastest growing cities in the state due to the increasing presence of medical companies, hospitals, and industry influence in the area. In fact, nearly 1/3 of Temple's workforce is employed in health-related fields. THBD's 5,000 square foot office and laboratory facility provides equipment for prototyping, testing, 3D printing, and cell research. We are one of a kind in the state. We support startups and small to medium-sized companies with ample resources, mentorship, and a wide network of collaborators. If you're interested in the opportunities awaiting you in Temple, I invite you to visit templebioscience.org to learn more. But for now, I'm so excited to introduce today's speaker, Brent Moody. Brent Moody has a corporate R&D role focusing on early preclinical to phase 2 development of small molecules and peptides for oral and injectable delivery. With over 24 years experience in the biopharma industry, he specializes in early stage drug product strategy and formulation development. Brent, thanks so much for joining us today. I encourage all of our attendees now to please mute your audio during the presentation and enter your questions into the chat at any time. At the end of his presentation, Brent will address these questions, and I'm going to be moderating those and sharing them from the chat. So with that, Brent, I'll let you take it away. Thanks again for being here.
Yeah, thank you, Ashley, and thank you to everyone for joining us today. I appreciate it. So, the title of the presentation today is Streamlining Your Drug Product Development Programs, and Catalent is we are what's called a CDMO, which is a contract development and manufacturing organization. So we provide formulation development and manufacturing of finished dosage forms for our customers worldwide. And so we're in a unique position. We see a lot of different things from a lot of different types of companies, a lot of different technologies, a lot of different molecules, and I think it puts us in an interesting position where we can hopefully comment and hopefully help give you guys some insight that'll help you guys as you move your new therapeutic drug candidates forward. So the agenda today, I'm going to start by talking a little bit about defining the difference between early and late stage development. What do we mean when we use those terms? We'll talk about why the formulations matter, how to assess the developability. I use the term developability, which is formulatability or deliverability of small molecules for oral dosing. So our company provides biologic support, injectable support for peptide small molecules. Today, to keep it succinct, I'm going to focus on oral delivery of small molecules, although I think some of the principles apply across the board. Also talk about selecting the right clinical dosing strategy for your molecule and the impact of a really high level look at the impact of those early formulation decisions that you make in that early stage. What do those decisions do to you from a downstream timeline and cost? And hopefully we'll have enough time at the end to take a couple of questions. So let's start with early versus late stage development. So I actually stole this image from a colleague. When the first time I saw it, I liked it so much. I said, I'm going to take this. And I really think it's picture's worth 1,000 words, and it speaks to not just technology development in the biopharma space, but I think in any technology. Early on, when you're faced with a lot of seemingly small decisions, that it's really easy to take the wrong path, and you need to make sure that you're on the right track. And that's kind of the focus there at the very beginning when you're in early development. And if you take the wrong track, it can have huge consequences for you down the road. Whereas in late stage, later on, once you're on the right path, it's all about making that journey as smooth as possible. Another way of looking at it is there are really two major stages of new technology development, and they require very different organizational goals, structure, and approach. So in early stage, an organization is really a truth-seeking organization. They're testing. You challenge yourself. You challenge your technology. You challenge your business theory. You're in pharma. You're establishing efficacy or lack thereof. If it doesn't work, you need to kill it quickly and move on to the next one. Whereas once you have established proof of concept and your technology works, then you switch gears and you become a success-seeking organization. You've demonstrated to yourself and to the world that there is value added in your technology, And now you need to think about how to get it to market, how to get it to the end users. Early on, these organizations are looking to reduce risk. They're loyal to the experiment, very scientific, very data driven, should be anyway. And obviously operating, there may not be any revenue at all. So you're obviously operating on a very low fixed costs and low capital requirements, small groups, everybody's wearing a lot of hats, and emphasize testing. So the folks that are in, in incubator settings, this shouldn't be any news to you guys. But later on, hopefully, once you've established your proof of concept, you're looking to maximize that value. You're focused more on getting it to commercialization. Roles become more specialized, or more people around, and emphasize refining the technology. And really, in drug development, we usually consider the line of demarcation between these two stages to be clinical proof of concept. So often, usually that's clinical phase 2. Phase 1 is safety. Phase 2 is safety, but we begin to look at efficacy and demonstrate some clinical proof of concept. And then, of course, late stage is stage 3, and that's leading towards commercialization. But once you've established that clinical proof of concept, in historically, your probability of launch of commercialization and launch goes way up. And so, if you're in an incubator setting, you're probably not quite there yet. You're somewhere else. You're probably more familiar with this slide, which is, should be very familiar. These stages should be very familiar to folks that are in drug discovery and research. I use this to talk about, where are you at, and where does Catalent engage and help you? And, discovery scientists are very familiar with establishing disease biology, finding targets, screening libraries through high throughput screening assays, confirming with second hits. And then as you get into lead molecules, or begin to identify multiple leads or multiple analogs of leads, you get in, then you start to think about the toxicology and the drugability, or the formulatability, or the developability of these molecules. And then you start to think about testing for pharmacokinetic properties. And really, that's usually in mice for the first time. And that's really where Catalent engages. And that's where the rubber hits the road in terms of delivering a therapeutic drug candidate to a living species. And usually that's when we get our first phone calls. And I call this late stage discovery. And the reason you're talking to someone like Catalent or any formula, or talking to your formulation folks at this point, is this because now suddenly formulations start to matter. So why do formulations matter? You can run into all kinds of issues when you start dosing animals. And this is dummy data. But it's taken from it's based on true stories, and it's the common things we see a lot of times. So ideally, you see on the left graph, you have for example, a 5 milligram IV dose, and these are if this was your profile. What you'd love to see is an oral dose profile that is similar, close to the IV profile, with minimal variability. But it can be surprisingly difficult to achieve. And often we see low relative bioavailability when dosed orally, and often with different rates of elimination, and in high variability and different rates of elimination. To make matters worse, sometimes you can sometimes see differences in species, which can just further confuse the issue. Why are we seeing differences in rats, dogs, maybe you're in monkeys, maybe you're in mice? And this is where you start to turn to formulation and to solve these problems. So I want to pause for a minute, and that brings up the question to talk about what a drug is actually seeing, or the environment it encounters once it is ingested. And the small intestines, you see on the left, we see the general outline of the transit time, the surface area, the pH ranges that the drug sees as it moves from the stomach into the proximal small intestines and into ultimately into the colon. And what you have to remember here is focus on the duodenum and the jejunum, because that is where the surface area is at. That is where the bulk of absorption will occur. And for a drug to be absorbed out into from the intestines into the intestinal wall, it has to be in solution. So the drug has to be soluble and in solution at this place. And so often you run into problems where a drug is soluble in the stomach, but if it precipitates out as it moves through, it will not typically be absorbed to any reasonable degree in the stomach. As it moves through the pylorus and into a higher, the pH range changes relatively quickly, you can see precipitation out. And so on the right, we actually took this graph from one of our spray dry dispersion presentations, but I think it makes the point. You see here an example. Often, if you have a crystalline drug that has poor solubility, you may not be able to achieve solubility above a minimum effective concentration to give you above a minimum effective concentration. I mean, if you do, and the example here is the amorphous drug. So if you look at the amorphous drug line, and you could easily replace the word amorphous drug with drug in solution. Maybe you've pre-dissolved the drug using a solvent like DMSO, which we often see discovery researchers use. Maybe you pre-dissolve the drug and it goes into the stomach in solution, or maybe it's a weak base small molecule that has pretty good solubility at the low pH of the stomach. Often what will happen is that will precipitate out as it moves into the proximal small intestine. So what you're looking to see is you need a drug that's going to stay in solution, either be soluble or stay in solution as it moves through the gastrointestinal tract. And in this case, you may need, if your drug is not soluble enough, you may need to utilize formulation to help it. Now, one of the things you're going to want to consider early on also is important is mode of action. And the reason being is, the mode of action, it could depend. What does it depend on? Does it depend on the area under the curve? Does it depend on a high Cmax response? Or is it total time above a minimum effective concentration? And it's important to know which of these in designing a dosage form and a formulation strategy, as there are options to alter those profiles with careful formulation. So how do I assess the developability of a small molecule for oral dosing? And so, in other words, you may ask this question: how do I know if my drug, if solubility enhancing technologies are going to help my molecule? And we start at Catalent, we start by using what is called the developability classification system, or DCS for short. Now, you've probably heard of the BCS, bioavailability classification system, which was introduced over 25 years ago. And it's you hear it throughout the industry, but it's important to remember that it was designed as a regulatory tool to support bioequivalency waivers for generic products, and it was quickly adopted by formulators as an easy way to categorize compounds. So compounds are put in one of four quadrants based on solubility, low or high, to the low to the right, high to the left, and permeability in the gastrointestinal tract, so low or high. And ideally, you'd love to be in quadrant 1, which is a soluble drug and highly permeable. And these drugs are relatively straightforward to formulate. BCS class 2 compounds are poorly soluble, but if you can keep them in solution, they should be readily absorbed from the small intestines. Of course, class 3 compounds tend to be polar and have good solubility, but they have poor permeability. And of course, class 4 are challenging. The problem with the BCS is that it tells you what the problem is, but it doesn't really do a good job of telling us how to fix the problem. And that's where the DCS comes in. So about 10 years ago, there was a paper published proposing a new classification system really for formulators. And it was an expansion upon the concepts of the BCS. So it builds upon the BCS with a couple of key advances. And one, the y-axis is still permeability, but ideally we want to graph it according to a predicted human PF, or PF in humans. And that's typically obtained through Keiko studies, although we have software that can make relatively accurate predictions about this. So that's our y-axis. The x-axis is the important step forward was thinking not about solubility alone, but really solubility where it matters. So we want to know what the solubility of the compound is in ideally simulated intestinal fluid, fasted or fed state, but we like to look at fasted state intestinal fluid. And more importantly, it's really, we want to graph dose divided by solubility, not solubility alone, but dose divided by solubility. And it introduced the concept of a dose to solubility relationship. And I'll talk a little bit more in a few minutes about what that means. So the paper goes on to propose a way to calculate we call it SLAD line, solubility limited absorbable dose. And that's in the small intestines. And the point being is that at a given PF and a given solubility, as your dose increases, you move to the right. As your dose lowers, you go to the left. And there's at some point where you can estimate that anything above a given dose, the SLAD, will be that any API dosed above that level, in theory, will either not dissolve or is likely to precipitate in the small intestines. Now, the human GI tract is obviously much more dynamic than that, so you would never write this in stone, but it gives you an idea of if your plan is to dose above the SLAD line for your compound, you start thinking about you're now in what's called a truly solubility limited space. And you need to think about solubility enhancement technologies. Whereas if you're in what's 2A, we're able to bisect class 2 compounds with the SLAD line. If you're in class 2A, you really should be soluble, but you may be dissolution rate limited. So those strategies are sometimes I would call maybe a little less technology advanced, but still requiring some manipulation, maybe micronization, initial solubility enhancement, but the drug should stay in solution as it goes to the intestines. So using that, so thinking about that, what does that tell us? And what it tells us is that you can adjust your formulation strategy depending on the highest target dosage strength, which you may not completely know, but you hopefully have some idea from the potency. And this is, so as an example here, we have a drug at a given PF and a given intestinal solubility plotted at a 5 milligram, a 50 milligram, and a 500 milligram dosage strengths. If you see here, the 5 milligram should be relatively easy to formulate. And even at a 50 milligram, while we're close to the SLAD line, you may want to suspend it early on, or add a surfactant, or maybe you consider a better salt form to get it in solution. And that may be the extent of your formulation work or your sophisticated formulation work. Whereas if this particular drug, if you're at a 500 milligram dose, there's a reasonable chance that even if it's in solution in the stomach, a significant portion of that dose is going to precipitate out in the small intestines as it passes through and it will ultimately pass through the GI tract undissolved. Another advantage to the DCS is it gives you a good reminder. If you look at it in different fed and fasted states, we use fed state intestinal, simulated intestinal fluid, and fasted state intestinal fluid. It gives you an idea of the potential for a food effect, positive or negative. And we can see when there's a significant difference in fed versus fasted, we may want to consider formulations that can leverage that difference and or smooth it out so we don't have problems with that. Okay, so what can you do if you're a solid DCS 2B compound? What are my options, and what are the technologies that we're talking about? And there are a number of technologies that are common to the pharma industry that are used throughout the pharma industry to enhance solubility in the gastrointestinal tract. But when selecting a formulation strategy, it is important to consider technologies that kind of check certain boxes that could have huge impacts down the road. For one, you want to have proven technology where the mechanism is understood and to some degree predictable for your molecule. You want technology that is scalable and supported by equipment and excipients that are well understood and readily available under GMP standards. And at the end, you want to have technology that produces a patient-friendly finished dosage form, something that's elegant, palatable, easy to administer, and stable for transport and storage. And really, in the industry today, there are really four basic technologies that I put in 3 different categories that easily fit this description. And so on the left, you see 2 technologies that are both fall under the heading of amorphous solid dispersion. So with either the concept being that you render what would typically be a crystalline API, you render it amorphous and you stabilize that amorphous form. The problem is most of these APIs will convert back to a crystalline state, so they will not stay that way. So you want to render it amorphous and stabilize it that way through the use of a pharmaceutically acceptable polymer. So you create a amorphous solid dispersion, or ideally a true amorphous solid solution of API and polymer that is amorphous. And the polymer serves a second purpose, which is to not just stabilize, but also once in solution, they are concentration sustaining polymers. So we being amorphous gives you that. Go back to that graph we call it spring and parachute effect. We can get the spring of solubility for a fully soluble drug by being amorphous, but rather than just precipitate out, it will stay amorphous, and you get aka the parachute effect, and it stays in solution. But because the polymers provide that concentration sustaining power. And so we do that through spray dry dispersions and hot melt extrusions. And I can do whole presentations on the technology, but just suffice to say that these are commonly used. We use them, and they're used throughout the industry. The second bucket is lipid-based drug delivery system. So in a lipid-based drug delivery system, you are solubilizing the drug in a lipid or like medium or long chain triglyceride or a lipid-like excipient or lipid-like surfactants in some cases. And so the idea is to actually pre-solubilize the drug. But again, these are typically self-emulsifying or self-microemulsifying formulations. So once they hit the stomach environment, they form micelles. And this serves similar to the polymer in the amorphous solid dispersions. It can keep the drug in solution. Now, I mentioned food effect earlier. One of the advantages of these is when you have a digestible lipid formulation ingredients, you kind of bring your meal with your dose. And so you can sometimes leverage your positive food effect as well. Particle size reduction is a third category, and here we're micronizing, and we can micronize, we can co-micronize with surfactants if that helps. And the idea here, the concept here is the important thing to remember is that with micronization, it really still is a dissolution rate enhancing technology. So more applicable to 2A compounds, although if you're in the neighborhood of the 2AB compound, it may suffice. And just, all of these are proven and scalable, available as GMP, and they have global reach and produce patient-friendly dosage forms on the back end. So I've talked a lot about dissolution solubility, but of course there's more to bioavailability than just dissolution of the drug in the intestines and being available for absorption. In this graph, you can see percent F, the final bioavailability, is a function of the fraction absorbed, FA, which is a function of solubility and permeability, and that's the DCS, but it's also a function of the amount of drug that survives gut wall metabolism, if present, and hepatic metabolism, i.e. the combination of those being the first pass loss. We have a lot of tools. We as formulators have a lot of tools to fix FA. We can get the drug in solution. We can keep it in solution. We can even sometimes help with permeability with certain types of excipients. But there's not a lot we can do. There are some things, but not a lot we can do with metabolism. It is an inherent property of your molecule. So we need to be careful and understand the drug and make sure that whatever that percent F is, we're not trying to chase or fix something that can't be fixed with a formulation. So how do we determine that? Well, we turn to physiologically based pharmacokinetic modeling, or PBPK modeling, to help us understand what is ultimately limiting the bioavailability of your molecule. PBPK takes into account solubility and permeability, like the DCS, but it also takes into the factors that the DCS does not, like efflux or active transporters, metabolism, and ultimately clearance. And it pieces them all together using computational modeling on a per species basis. And the idea is to let the software adjust the compartmental model to provide a best fit to the data. Now, at Catalent, we use the GastroPlus simulation software, and which is certainly prevalent throughout the industry, and we consider it the gold standard in the industry. But there are actually quite a good deal of modeling software available out there. And really, this is in addition to giving us or helping us understand what we need to be approaching as formulators. It can also actually help in candidates in candidate selection. And I'll show an example of that in a moment. But we're building these models to help us predict how given prototypes, and we produce prototypes from those different technologies, that we want to be able to take the PK data that comes out of that and use that to predict how those prototypes are going to behave in other species and ultimately humans. So here's an example of applying the PBPK knowledge to molecule selection. And here we have 5 example molecules, again, dummy data, but based on true stories. And if you were looking at potency alone, you would say number 2 is the clear winner. It's your most highly potent. A lot of times that's how researchers and drug discovery folks think. They're looking for potency. And it has similar, and if you look at it, it has similar bioavailability to the other potent compounds. So why not go with number 2? And I would say, let's look at the DCS situation and the first pass loss. Number 2 and 4 have similar first pass losses. But path number 4 is a DCS 2A, whereas number 2 is a DCS 2B. So we may be able to achieve solubility in the small intestines with something simple like micronization, or maybe just the addition of a surfactant. Whereas if it's a 2B, depending on how far it is on the 2B side, we may have to look at more advanced technologies. So on the right, if you look, while both of these without any enhancement are only giving you about 33 to 36, 33 to 21% bioavailability, they will, if you can maximize the fraction absorbed, they both are equivalent. And the point being is that molecule 4 is a lot easier bar to jump over than 2. So in this case, I would actually say 4 is the winner. So with all that in mind, how do we select the right clinical dosing strategy for your molecule? And I would say the number one rule for that is think about your patient. Don't forget about your patient. And this starts by thinking about not just the indication, but also the mode of action. And what the indication tells us, it tells us something about my patient group. Are they going to be adult healthy volunteers, or are they going to actually be actually target patients in the first in human studies? And those patient populations could be pediatric, so they may be very limited in what they can take from an oral standpoint. They may be geriatric, maybe high pill burden, trouble swallowing. They may be, typically they're often, if you're in patients in first in human, it's probably oncology. It's often oncology, where again you have other drugs in play. You may have, again, pill burdens, and again you may have trouble swallowing. So there may be a big difference in the overall approach to the finished dosage form depending on those factors. But we also want to think about the mechanism or the mode of action, because it could be the interaction of a drug with a receptor, an enzyme, a chemical or protein, or it could be acting, or it could be just acting locally in the gastrointestinal tract, and absorption is actually not required. The important thing to remember is that the mode of action determines where the drug needs to be in the body. And this is not necessarily obvious from the indication alone. So the point is that there are important considerations that we want to share with your formulation team. You have these conversations, or the formulation team should be certainly aware of these and thinking about these as they help you develop your formulation strategy. So with understanding of the patient population and overall clinical strategy, we can move to selecting the right dosing strategy. And what I put up here is a not necessarily exhaustive list, but it's a fairly thorough summary of the typical early stage first in human dosage forms that we see: drug in bottle, drug in capsule, liquid in bottle, liquid in 2-piece hard shell or soft gel capsules, formulated blending capsules, or maybe even tablets where you're using either IR excipients or spray dry dispersions, HMEs, delayed release, sustained release, and coated and uncoated. So I want to go in a little bit of detail about these, so we can address a little more, going roughly from the simplest to the most complex. Drug in bottle. Maybe you've heard that term, drug. What is drug in bottle? What does that mean? Essentially, it's neat API. It's just crystalline, often crystalline, maybe amorphous, often crystalline API shipped directly to your clinical pharmacy, and it's either dissolved, either reconstituted or suspended or diluted for administration directly. So the advantage to this is that it gives you a lot of flexibility in dose, and it allows for extemporaneous preparation, so you don't have to go through the GMP manufacturing process. You can have a clinical pharmacy dose and administer it. There are challenges to think about. You have a taste that, maybe there's a gag reflex, or if for some reason you need a placebo in the first in human studies, you may have to think about placebo masking. Can be challenging with taste. And you may still need, depending on the solubility situation of the molecule, you may still need to think about things like micronization or the addition of diluents or acceptable diluents that may get it in solution. So it may not be without formulation requirements, and also may require precision weighing by a third party, by the clinical pharmacy. So you just want to make sure that they're able to handle that. Kind of the next more complicated strategy would be drug in capsule, and some people call this API in capsule. Some people call it PIC, powder in capsule. I think the industry has begun to move to clarification that it's neat drug in capsule, and that can be done a couple of ways. It can be done through precision weighing into hard shell capsules, which is one of our pieces of equipment on the left. This is good for when you have very low dosage strengths that may be difficult to precision weigh by the clinical pharmacy. The important thing to remember here is you just have API in a hard shell capsule, and there's nothing to help get it in solution or certainly keep it in solution. So a better fit for those DCS class 1s, maybe the 2As if you're using a micronized approach or a better salt that may work for you. It's a less flexible than the powder and bottle, so you may have to make a couple of different dosage strengths to give you the range that you're trying to achieve. and they can be prepared by hand. And on the right, we see a flood fill strategy, which is less accurate than it's less accurate than the equipment, but it certainly would typically able to be very precise with the flood fill strategy. So, and the same considerations apply. Now, liquid in a bottle is another way you could go by this. I separate this from drug in a bottle. Where drug in a bottle, we send API to the clinical pharmacy and they reconstitute it there. There may be reason to take a liquid in bottle approach where it's prepared ahead of the pharmacy and it arrives as a solution or a suspension. And often this is we can take the lipid-based delivery systems can take this approach. Again, it gives you high flexibility of dose. Taste and placebo could still be an issue, and it does require some development and analytical support. It may you need to establish the physical and chemical stability of this dosage form. And it can be produced in bulk for sampling or precision filled into single dose units as well. Now, the liquid filled hard shell capsules, this is a lipid-based delivery strategy where you're pre-dissolving, or in some cases, lipid suspensions in a lipid vehicle and precision filled into hard shell capsules. And this is a case where we're looking for bioavailability enhancement through improved solubility and even perhaps permeability. Some of the lipid excipients help us leverage, and we can move compounds that are maybe in the DCS class 4, maybe even the DCS class 3 into a class 1 or 2A to a 2B situation through permeation enhancement. Again, it's the same thing as the powdering in a drug and capsule, in that you have a little less flexibility. You have to have multiple dosage strengths, and these can be prepared. These can be filled ahead of time and sent to a clinical pharmacy, or in some cases, you may be able to send the bulk and have the pharmacy fill them. And then, of course, these hard shell capsules, if you start this strategy, you're heading towards a soft gel strategy. Ultimately, in the long term, for scalability and economy and manufacturability, you'll move towards a soft gel, and that's just another way of forming the liquid piece 2-piece hard shell. Finally, we have formulated blending capsules. I like to use the term blend-in capsule, and that blend could be a blend of just API blended with certain immediate release functional excipients, or it could be a spray dried dispersion or a hot melt extradate that has been either granulated or milled and blended with other excipients that filled into capsules. So more of a formulation strategy. It can also be something very simple, like a binary blend of API with a filler that provides disintegration to help with rapid dissolution, or maybe a surfactant could be blended in. And then finally, you have tablets. So often don't see these in clinical phase 1 because of scale. The API may not be available to scale this up, although we can work with actually very small amounts. But this is a lot of times these spray dry dispersions and the HMEs work better as tablets. You may want to put functional coatings on them, delayed release coatings. So a little bit more of a sophisticated strategy there. And so to wrap this up, I've summarized these various strategies, and this is a busy slide, and I'm not basically repeating what I just said, but I wanted to draw attention to the DCS fit for each of these. And depending on where your DCS situation is, I would say you evaluate your DCS, but then you back that up with PBPK modeling and use that as essentially, this is a guide to say, this is the strategy we need to go down. And so finally, what we're usually trying to do when developing a strategy for a new drug product is essentially to populate this type of a table with our customers. We try to sit down with the customer and understand, make sure we know as many of these answers as we can. We want to think about the indication and the mode of action. We've talked about why that's important, how often and how much we need to dose, what's important about the PK profiles, need to be area under the curve, Cmax, time above MEC, but also how many doses. We don't just think about supply of the product, how many doses are needed, when and where do they need to be, and most importantly, what kind of a patient is taking them. So the impact of, once all this is taken into account, what's the impact of these early decisions on your downstream timelines and costs? And really what the question we get from customers is basically boils down to, how soon can I be in the clinic? And so I wanted to give you a sense, I want to put up a sense of timeline, give you a sense of the relative difference in timelines of the different solubility enhanced dosage forms. I thought that might serve here. So I'm presenting these timelines with a huge qualifying statement that no two compounds are alike, no two projects are alike, no two customers no two clients' business situations are the same. No two APIs are alike. And these are I think these are very conservative timelines. So this is from once the time we've selected the right strategy, how long is it going to take us to have a bottle of dose units on a clinical pharmacy ready to be dosed into patients, labeled, released, package labeled, released, ready to be dosed. And, and these are conservative timelines, and there are ways to accelerate them, but it really will always depend on the drug. I have intentionally left off costs. And for one thing, I'm not at liberty to give quotes without a CDA. And honestly, it's dangerous to throw out big numbers without looking closely at the drug. But I would say roughly there's probably a 2x cost difference from the first, more simpler, faster of these options to probably the more complex option. And there may be ways there's even, if you think about it, there's even this doesn't include the powder and bottle strategy, which could get you maybe there even a little quicker. So it's important to really, the take home message in picking any of these strategies is, first, know thy patient always, but know thy molecule, know your clinical and your business strategy, and use that knowledge to plan ahead. And certainly don't be afraid to ask for help. We are happy to always talk with customers who are future customers who may be still a long ways off from working with us or formulating drugs, just to start thinking about those very early things that we talked about. What is the more druggable? What are the druggable aspects we need to take into account, and really use that to plan ahead. And we're always happy to have conversations with you guys, and hopefully this will be something that you can take back and look at your program. So with that, I will pause, and I will take any questions that anyone may have. And I thank you for your time, and I hope this has been helpful.
Brent, thank you so much. This has been wonderful. I really appreciate all the work that you put into it. Now's the time, guys. Brent is a captive audience. Ask your questions. Don't be shy.
Brent, can you tell us a little bit more about what it looks like to work with Catalent and kind of the steps on getting the conversation with you to start this project, start this discussion?
Yeah, I mean, honestly, I would let my colleague Elaine answer that question. I mean, usually the first person, she's our account director, and she's usually the first person our customer talks to and says, "Hey, I've got something I want to talk." So I usually talk to that person. Elaine, you have any comments on that?
Oh, we can't hear you.
You're muted.
Sorry, I was double muted. So yeah, thanks, Ashley. That's a great question, and I can help with that. So as Brent mentioned, typically, we will engage with customers early on the business development side. So my role at Catalent is business development, and I'm responsible for maintaining and establishing new and existing business relationships. So oftentimes we will have introductory calls with someone who may have a project that they're interested in, and talk with them. They may either come through from maybe a webinar they've seen at Catalent, or maybe through our inside sales group. And then we have a preliminary discussion set up, typically like a confidentiality agreement. And then we have a technical call after that with Brent and the S&T team. So we do have, as Brent being part of the science and technology team, we're able to kind of vet projects through them, and they're able to help triage, which site might be the best fit for projects and such. So that's kind of the initial how we get started. And then from there, of course, if someone has a full blown, request for proposal and such, we'll actually loop in the specific site that would be supporting and that technical team as well.
Yeah.
So
go ahead.
I just have a quick question. So at what stage do they contact you? At the idea stage, or do they contact you at the point, hey, the compound's working on my models in vitro, or do you wait for in vivo?
Yeah, so it can really be at multiple stages. Where Brett has highlighted here is probably the best spot to get started. We do have our site in RTP that can also support with characterization of API and such as well. But once they have identified a lead compound, then that's usually the point at which we get engaged and we can start having some conversations around how we could support.
Now, I will say that what we'll do occasionally, and by occasionally, I mean, probably, not that, actually relatively often, is customers will have a scaffolding, and they may have a couple of lead candidates from the scaffolding, and they're not quite ready to isolate those leads, and they're not quite ready to have enough to really start going into animals with it. But they hopefully see the value. They see the value in the DCS classification, and just kind of getting an idea of what I say with the DCS. It doesn't tell you exactly where, but it tells you what ballpark you're playing in, and so we will take structures. So of course, the CDA, of course, in place. We'll take a structures, or maybe a couple of structures from a series of compounds, and we will run them through our GastroPlus software. And by run them, I just say put the structure, and GastroPlus will provide estimates, in silico predictions, and I say estimates at best, of important things, the important things like permeability, which I actually trust GastroPlus to. I mean, it does a pretty good job of giving you a reasonable estimate of permeability, solubility in biorelevant media. I've seen it be off significantly. I've also seen it be on, but then you can kind of cross-reference that with what you know about solubility of the compound at different pHs, and other things like it will make predictions of potential for substrates. It will also, we can get an idea of the log P and the pKa, and we'll rank order those for our customers. So we'll put a little graph, a little table together. It says, this is compound A, B, C, D, E, F, and G, and here's your DCS situation based on this, and here's your log Ps, your pKas, and that's just a piece of data for them to go in, and we'll do that at a, if it's a reasonable amount, we'll do that at an extremely low cost, and it's something that they will sometimes use. Folks will use it, from their own. I think they've used it in grants. I can't speak to that, but I see they've taken a little PowerPoint presentation with the data and said, okay, this is this is why we're selecting this one of the reasons we're selecting this compound over that one. So we'll do that for customers as well. And that's I would call that that's about as early as we get involved.
We do have a question in the chat, if you'd like to address it.
I'm not seeing the chat. Will someone read it to me?
Okay, so you want me to read it?
Yeah, so without breaching CDAs, Carrie asks, does Catalent have any experience, and can they speak on hallucinogenic drug development, specifically new molecules, compounds, formulation?
Well, yes, in that. So we're agnostic to indication, therapy categories. Now that said, there are certain types of molecules that their mode of action, certainly, there are we see protein degraders right now, and that's a hot topic, and we see we see covalent molecules, which very long residence time on target, and that may change you from a formulation strategy. But my point is that you say if a drug is if a molecule is hallucinogenic, what does that do to me from a formulation standpoint? It to me, I'm agnostic. It's is it permeable? Where does it have to be? Where does it have to be? How long does it have to be there? And what as a formulator, what do I have to do to make that happen? Now, an important thing for us as a vendor is where can we, those are typically, they're going to be controlled substances. They're going to be scheduled and controlled. And so we do have to take, not every single site. We have a lot of sites that do different things. Some of them do the same thing. There's redundancy. And where can we handle those? So we may have to, it may impact what we can offer you, in that we cannot take that compound and put it in any of our facilities. So I don't know if that answers your question, but we would classify it. We have a team that goes in and looks at that and classify. It's an onboarding process at Catalent. So we'll have the conversation with you. We can work on them. We just can't work on them in every site. That answers it. If that was your question, I hope. I'm not sure.
And I could add to that from the business side, we have seen a bit of an uptick in, if you're referring to like psilocybin compounds, we've seen an uptick in those on the business side. And it is something that we've, we have a few programs where we have experience with that.
Okay, she says, cheers, that's helpful. So there we go.
Hopefully.
And And I don't necessarily have the most scientific background, but I would love to know a little bit more about how you're determining. So, for instance, with different drugs that come out, some are just some are do the animal testing, some do not, some do human testing. How do you determine that? What is the qualification?
Well, all new therapeutic drug candidates will have to go through a non-human safety testing round of non-human safety testing. The short term for that is GLP. We call it GLP tox. So any new candidate will have to be demonstrated safe at significant levels above what you would put it in a human. And usually it's a non-rodent species. You have to test in 2 species. You have to do ascending dose studies, and you have to demonstrate essentially what that therapeutic index is for your molecule. And that data is part of what goes into what's called an investigational new drug application, or an IND. So to to dose a new molecule into a human, you have to have an open IND. In the United States, the FDA, but there are other regulatory agencies around the world. And to get that, you have to have demonstrated safety in animals. So I'm not really sure. I'll never say never, because this is too diverse of an industry. Tammy may know more than I do on that subject, but you're not going to ever be able to put a new molecule into a human being without first having shown safety in animals. So animal testing, I'm a dog lover, so animal testing is not something I love that we have to do. But it's a necessary evil, because we have to prove that drugs are safe before we can go into humans with them. So does that answer your question? There's always animal testing up front. And Tammy, I don't know, maybe you can elaborate on that.
I agree with you 100%. At this point, where we are right now today, it has to go in animals first. And you know what? That's good for us as humans, because you don't want a drug that can cause convulsions, let's say, in a dog, be given to a human without testing it in the dog first. So, I mean, if it causes convulsions in a dog and it crosses the blood-brain barrier, it ain't going in humans. So it's an important science.
I appreciate the response. I think that it's interesting when you go into the beauty industry, for instance, and there's a lot more focus on how it was tested, but then again, beauty products aren't necessarily drugs. So it's a fine line, but it's interesting.
I do have a question. So Brent, let's say you have a compound that you're working on and it's only soluble right now at DMSO. If you take it and you put in any other kind of combinations, it's falling out of drug. So is that something that Catalent would handle for me and figure out what's the best diluent that I should use my drug with?
Yes, absolutely. We do that all the time. I mean, it's everybody knows, all the researchers use DMSO. They go grab it off the shelf, and that's what they use. Just it's a strong solvent, with toxic solvent. And so often that's one of the first conversations we're having is, my drug is only soluble in DMSO, and it's causing me problems. It's muddying my data. It's muddying my tox data. And my accent, you're gonna have to forgive it. And that's usually what the first thing we're looking at. So some of that early work we do is we do fit for purpose formulations. A researcher doesn't, you're not at a point where you need a human formulation. So you may not want to go as far as a spray dry dispersion or a hot melt extradate, which is equipment heavy, technology heavy, expertise heavy, and ultimately costs more. The question is, can I just come up with a solution, an alternative solution? Right. And Tammy, usually we're looking at that's more on a lipid side. We're going to take more of a lipid approach, mm-hmm. And it may be if it's not true lipids, and it may be lipid-like excipients or lipid-like surfactants. And we actually have some off-the-shelf technology. We actually have a formulation kit that we provide to researchers that has these pre-manufactured clear colorless non-aqueous. So there's no solution in any of them. There's no water in any of them. they're all, but they're lipids, they're fatty acids, surfactants, liquid surfactants, and pharmaceutically acceptable co-solvents. And a lot of times we will give that to a researcher and say, here, use this. And there's 8 different solutions in there that target different types of modalities of solubilization. And hopefully we see a pretty good hit numbers on getting that in solution. But the important thing is that's we call that our Optiform select kit. I just would want to have a conversation with a researcher to explain to them that is not a toxicology formulation. The levels of excipients in that are going to be too high to go do long-term studies in dogs and rats. In fact, these formulations are only targeted for rodents. And, but we can talk about how to use it and screen it. You need minimum quantities of API, but you can hopefully get a little better solubility without DMSO, and they're safer than DMSO.
So Brent, where were you when I had to take drugs, put them on a spinner with a magnet inside of it, spin it up until the minute that I am injecting into mice? I mean, seriously, where were you?
Yeah, well, that kit comes from years of it's actually not our first iteration of the kit. We built it over time. Like the very first kit many years ago was not as functional, but look, Tammy, the truth is customers come to us and say, well, hey, I've got to go in a dog for this reason or that reason, or hey, I also need an injection. I also need an injectable formulation. For various reasons, because that kit we have is for oral only. We can also do a rapid service in-house where we can essentially take small quantities of your API, do a quick solubility visual. So we're not even bringing HPLC in at this point. We're doing visual solubility screening of base types of excipients and co-solvents, complexing agents, and use that and spend a couple of days of designing a fit for purpose formulation. Again, not trying to make you a toxicology formulation, but something that'll just get it in solution and hopefully give you a potential for better, again, the challenge you're always going to have is unless we get into analytical and have some in vitro data, it's hard to really predict. Even then, it's hard to predict how it's going to behave in the animal. So I would just say that that's more of a get it in solution approach and hope that helps. If you have one of those true DCS2B compounds where all of that API crashes out, you may be true. You have to spend a little bit more time and get more of a demonstrate self-emulsifying formulation. So, it's tough to say, Tammy. We have to sit down and have these conversations with our customers in order to customize approaches sometimes of what works best for them.
Great. Well, thank you for that. I'm sure that was a question that other researchers were thinking about, because I know that was something that I had to deal with for years and years. Oh, wait, and years. There we go. Right. If there's no more questions, I want to thank you guys again. This will be on our YouTube channel. So if you want to re-listen to any specific part, we can go ahead and do that for you. Again, is there any other questions? All right, Ash, you want to close us out?
Thank you. Thank you, everyone.
Yes, thank you so much. Again, Brent, if you want to pop up that contact slide so people can know how to get in touch with you. We also welcome everyone to check us out on templebioscience.org or on social at TempleHBD. And you can see what we're up to, see about the latest webinar coming up, and get to watch this webinar and others in our e-learning series library. So thank you so much, Brent, for joining us. Thank you as always, Tammy, and we look forward to seeing you all at our next webinar.
Brent, Elena,